Hello student, morning to all. I am Dr. Tariq, Department of Biotechnology, Islamia College, Body. Today we discuss about microbial biotechnology. This is the last topic in this unit, the unit 2. So I have to study in each and every videos in my topic repeatedly, because next week the exam will start, CAA test 1, so that you have to watch the full unit in previous slides also. Any doubt, ask me to contact the WhatsApp in my personal number. Then what I said, the unit is keenly observed and you have to return the examination successfully. Okay, now molecular diagnosis of infectious disease of AIDS. So in previously the molecular diagnosis infectious disease of tuberculosis, in previously malaria. At the same time the topic is finished in the molecular diagnosis infectious disease of AIDS. So this is the main studies. A specific test for HIV infection. For example, there are 4 types. Antigen detection test, it's available in the market. P24 antigen and virus isolation test. Detection of viral nucleic acid test, and another one is antibody detection test. So that this is the HIV infectious specific test available in this market. Now, some of the non-specific tests are available. It's total and differential leukocyte counting. Because of the leukocyte counting is really low, it will be affected in the cells. The T lymphocyte subtest available in the one of the assays. It's also the how many numbers of subset cells affected in this person. And platelet counting, you know very well. The IgG and IgA level test, immunoglobulin G and immunoglobulin A level test, and finally the skin test for CMI. So these are all the non-specific tests. Now we going to the antigen deducting tests, specifically available in the market. Actually, the antigen detection test, after a single massive infection, viral antigen P24 and RT may be detected in blood about 2 weeks. That means the P24 antigen is the earliest viral marker to appear in the blood. Globin M antibodies appear in about 4 to 6 weeks, to be followed by globulin immunoglobulin G antibodies. If the infecting dose is a small example in needle stick injury, the process may be considerably delayed. At the same time, the appearance of P24 antigenemia and viremia followed by immunoglobulin M antibodies response coincides with the acute illness. This is the most antibodies response acute illness in affected person. The P24 antigen capture the assay, that means ELISA test, enzyme linked immunosorbent assay, which uses anti P24 antibodies as the solid phase and can be used for this. The test is positive in about 30% cases. It will be antigen detection test. Another one is specific test for HIV, virus isolation. Actually, the viruses present in circulation and body fluid with the lymphocytes or free cells. The viruses titers parallel P24 titers, being a high soon after infection or low and antibody bound during the asymptomatic period, and again high towards the end. The infectivity being highest in the early phase, and when the person becomes terminally ill, the affected person, this is the highest phase in this person viral isolation test. It's continuously the virus is present in many parts of the body. Particularly, it's isolated from the peripheral lymphocytes by the technique of co-cultivation of the patient. Lymphocytes with the uninfected lymphocytes in the presence of interleukin 2. And some of the another way viral replication can be detected by RT. Note that because of the risk involved, virus isolation is to be attempted only in laboratory with adequate containment facilities. Detection of viral nucleic acid. As the most sensitive and specific test, polymerase chain reaction has become the gold standard for diagnosis in all stages of HIV. PCR, there are two types of detection: DNA PCR and RNA PCR in the detection of the virus nucleic acid test. The antibody detection, another one is 2 to 8 weeks or 2 months for the antibodies to appear after infection and during part of this period, the individual may be highly infectious, known as the particular part of the period. The infection can be detected during the path period by the P24 assay. Some of the antibody tests will have to be done after 2 to 6 months to ascertain whether infection has occurred or not. It will be possible. Immunoglobulin M antibodies disappear in 8 to 10 weeks, while immunoglobulin G antibodies remain throughout the days. At the same time, the specific test for laboratory diagnosis of HIV infection. The test may be P24RT antigen test. The test will be window period positive acute infection, also the positive symptoms. Some of the asymptomatic infections negative test. ARC and the AIDS test positive, so that the clearly the test will be all kinds of window period and the acute infection AIDS also. Another test is viral isolation, so that the window period is double positive tests. Acute infection is more or less, it may be positive or negative, plus or minus, it's available. The symptomatic infection is negatively available. AIDS is a positive test. ELISA is the window period is negative, and the remaining of acute and the symbiotic and the AIDS is a positive test. Western blotting test also the same one, so that this is the specific test for laboratory diagnosis of the HIV infectious person. And some of the screening tests are available: ELISA, rapid test, and simple tests. Some of the supplement tests is Western blotting test, indirect immunofluorescence test, radioimmunoprecipitation assay also available in these supplementary tests. And finally, the some of the tri-dot HIV kit are available in the markets. In India, not possible in this manner, because particular country will produce in this particular kit. It is very olden days. I think it is in 2013. It's available, produced in the market. This kit is triple dot, that means immunochromatographic assay. Immunochromatographic assay. This is a new one step. Rapid assays, also known as immunochromatographic assay, or convenient self-contained tools for HIV serologic testing. It's consisting of the flat cartridge device, usually plastic or paper. That means the whole blood or oral fluid or serum is placed in the tip of the device and allowed to diffuse along a strip that is immerginated, impressed with reagents, often protein A, colloidal gold, that bind the permit viral detection of HIV antibodies. Some use third generation antigen sandwich technology. It is another ISA test. These tests can be completed in less than 10 minutes, some within 2 minutes also. It's required a little or no addition of reagents, and it's contain a built-in quality control reagent to control for technical error also. Some of the tests can be stored at a wide range of temperatures from 15 degrees Celsius to 30 degrees Celsius. Transported easily. For example, one type determined about comes in curds. The curds require no reagents, just addition of serum or plasma. At the same time, other rapid tests formats include deep sticks, in which antigen is attached on the teeth of complex device. Several of these rapid tests have the ability to differentiate HIV-1 and HIV-2. So the tri-spot HIV rapid tests available in the market, but here it's not possible because of the temperature is one of the reasons. At the same time, HIV resistant testing also available, genotypic as well as the phenotypic measurements. The genotypic measurements, sequence analysis of HIV RNA genome is compared with known HIV RNA genome. Sequences having known resistance to drug, as well as the phenotypic measurements, growth of viral isolates of the patient compared with the growth of reference strains in the hands of the expert. Resistance testing enhances short-term ability to decrease viral load by 0.5 log compared with changing the drugs merely on the basis of drug history. So this is the HIV resistant testing, both genotypic as well as the phenotypic measurements. So that I conclude this is the most importance of in futures also. The HIV is a molecular diagnosis of the infectious disease of AIDS. So that I thank the watching the slide. Thanks for